All right, I want to pick up where we left off last time. This is a view of how far we got in the previous video as we introduced the different parts of the kidney that we're going to be talking about. And number one up for today is what we show there in pink. It's filtration. And so we need to understand Bowman's capsule. We need to understand the glomerulus, to some extent the juxtaglomerular apparatus, although that will be covered in a second video. Really, it's all about filtration. Understand what's going on in the glomerulus. So just to highlight this point again, if we come down here, we've got the inside of an endothelial, the capillary bed that's here. Now let's remind ourselves, Bowman's capsule is the part of the nephron that is receiving the filtrate. Filtration is all about getting through the fenestrated capillary bed to enter into Bowman's capsule. So we remind ourselves that we've got afferent arterioles, and then we've got efferent arterioles. And both of these can regulate aspects of what's going on in filtration. But what we want to do right now, first of all, highlight the three main effectors for filtration. What are the three parameters that affect filtration most? And we can list those as one, pressure. And arguably this is one of the most important. Number two, size. And number three, we can filter based on charge. All right, as we consider filtration, and especially pressure, but also size and charge, we need to understand what we're going up against. Our goal right here, if we were to actually add Bowman's capsule to this structure, Bowman's capsule would be sitting out here. And as this structure right here represents our endothelial cells, we recognize we've got to move through the endothelial cells, and our ultimate goal is to get over here into Bowman's capsule, where we would then join the filtrate as we enter the proximal convoluted tubule. Now, to understand how to get across the filter, let's first define the filter. As we look at this structure here, we're going to call this the filtration membrane. And we want to take just a second to appreciate some of the structures that we have in here. First of all, this funny looking cell right here called a podocyte. The podocyte provides barrier filtration. Now, how does that work? Well, take a second to appreciate what the podocyte looks like. Podo meaning foot. You can see these foot-like appendages that reach out from the podocyte. These will actually intercalate or interdigitate with one another to provide a really powerful barrier. And we can show an example of what podocytes essentially holding hands looks like. If we take in a close-up look, we can see these two podocytes here and here, and how they interdigitate, providing this filtration membrane, or part of the barrier filtration. The gaps in between these podocytes serve to provide structural support. And thinking about a coffee filter, although maybe that's not the best thing for this group, filtration based on size. So the podocytes are a key part of this. We have another cell type up here called the mesangial cell. The mesangial cell is a support cell. Recent evidence has showed that it is contractile, so it can squeeze or it can relax, and in doing so, change the overall dynamics. It's not super important that we go into understanding that. What else do we see in here? What about these green lines? This is the basement membrane that these endothelial cells are attached to. And the basement membrane is actually really important in this process, because the basement membrane in this area is unique in the fact that it is built with a lot of negative charges. Now, I want you to appreciate the value of that as we discuss charge in just a minute as a key regulator of this process. But let's finish labeling some of these other structures. Within the endothelials themselves, we see these structures here called fenestrations. Fenestrations are essentially a hole that goes right through the cell membrane, and it has a diameter of about 70 nanometers. And so this is another way that I can filter based on size. So We see these limitations built in the filtration membrane. We see limitations based on charge. We see limitations based on size. But we got into this talking about how pressure is the most important and the one that I can regulate the most in dictating the overall filtration rate. To understand that process, it's first, I think it's important to remind ourselves, and we've talked about this already, the concept of hydrostatic pressure. When we consider hydrostatic pressure, we've learned that resistance is proportional to 1 over the radius to the 4th power. In other words, resistance increases significantly as the overall diameter decreases. And I can use this to my advantage as I regulate hydrostatic pressure coming into and out of the endothelial cells. I think it hopefully makes common sense that the higher the pressure, the more filtration is going to happen. The more I can force fluid, plasma, and its dissolved contents to leak through these fenestrations and past the filtration membrane, the barrier set up by the podocytes, the barrier set up by the negative charges in the basement membrane. Increased pressure will allow me to force fluid through there. And this provides a good opportunity for the body to regulate what's being filtered and what isn't. Now, a couple of things to make note of. Number one, the hydrostatic pressure in this capillary bed is higher than what we've normally seen. If you remember the hydrostatic pressure entering a normal capillary bed for capillary exchange, it was right around 30. Here, we're pushing around 55 millimeters of mercury. Now, it's important to recognize I can go that high without damaging the endothelial cells because of the structure built in, right? Remember the podocytes. We talked about them providing structural support. That's what allows me to increase the hydrostatic pressure. Without the structural support surrounding these endothelial cells, that type of pressure would rip them apart. So it's important to be able to recognize this so that I can maximize filtration without damaging endothelial cells. How do we do it? Well, we've got two different things that we can do. In order to increase the filtration pressure, I can increase the radius of the afferent arterioles, or, and really it's not either or, it's a combination of both, I can decrease the radius of efferent arterioles. Now, just to give you a sense of what that looks like, all right, we've got the afferent arterioles coming in, and then we'll just kind of expand this. This is the glomerulus where we've got exchange happening. If I increase, looking at this first one, if I increase the radius of afferent arterioles, we'll label this afferent. Let's expand these walls so that now we're a lot bigger. Well, what have we done? We've increased the volume, the flow, significantly. Because we've decreased resistance, we've increased the flow. And that allows increased volume. And of course, if I've got increased volume, I'm going to have increased pressure. It's just like putting more water in a water balloon. Please note that I did not change my efferent arterioles. If I would have concomitantly increased the efferent arterioles, then the increased flow coming in would have been counterbalanced by the increased flow leaving, and the volume inside the glomerulus would have stayed the same. And so I can increase the afferents while keeping the efferents the same, or you can appreciate we'll get the same effect if we decrease the efferent. If we make that a really small hole coming out, now what are we going to see? We're going to see increased volume inside the glomerulus itself, and that's going to drive an increase in pressure. So let's come back to our picture where a typical hydrostatic pressure in the endothelial cells is going to be right around 55 millimeters of mercury. Let's see if we can follow the process. And we want to look maybe at two different molecules to understand size, to understand charge. Let's compare and contrast urea, which looks like this. Of course, those amino groups have lone pairs of electrons, which is going to be important. So there's urea versus albumin. Albumin, as you remember from our blood unit, is the most abundant protein in the blood. And as a protein, number one, it's going to be a lot larger. And number two, what's it going to look like? It's going to look like this. I think that's a pretty good description of albumin. Notably, recognize that albumin and most proteins in the blood carry negative charges. But let's look at urea on the contrast. Because these nitrogens can act as organic bases, they can actually pick up an H plus here, an H plus here, and this gives urea a strong formal positive charge. So as urea comes into the blood, number one, it's small. Comparative to proteins like albumin, it's small. Number two, it's got a positive charge on it. That's going to allow it to pass through these fenestrations, and it's actually attracted to the negative charges in the basement membrane. It wants to leave the blood. And as it gets pushed out, it just gets flushed through the system. So urea is something that's going to go right through. But if we contrast that with albumin, the size in general prevents it from easily going through those endothelial cells. Plus, because it's got a negative charge, it's repelled by the basement membrane. And so we shouldn't ever see a lot of albumin inside the blood. Albuminemia, inside the urine. If we see that in the urine, that's suggestive of damage to the glomerulus and problems with filtration. So, in summary, filtration is dictated by these three key processes: pressure, size, and charge. Size limits are placed based on the fenestrations in the capillaries and also the barrier filtration driven by the podocytes. We can see that down here. Charge is primarily associated with the negative charges on the basement membrane that repel negative proteins from getting through. And then pressure, which is the body's main way of regulating glomerular filtration rate, GFR, is dictated and regulated by the radius of both my afferent, primarily my afferent, also to some extent my efferent arterioles, where increasing the radius of the afferents and decreasing the radius of the efferents drives increased hydrostatic pressure and therefore increased filtration rates.